Hello everyone, this is Dr. Rafael Gutierrez, and today I'm going to talk about the basic anatomy terms, cell biology, and tissues. Now, if you're taking anatomy as a class, this should be a complete review. The cells are something you should have covered in general bio, and tissues we'll talk a little bit about. Basic anatomical terms are also something that may be a little new. Now, the big thing that I would tell you to do is start making a list of what terms are. So when you see them again, which you will see most of these terms over and over again throughout the anatomy semester, that you have something that you could jump to directly to know what the different words mean. And that's actually one of the big things. Now, I will tell you that there are some things I usually recommend people do when they are starting to take anatomy. One is there is a link I'm putting below on the description on it's a KPBS story where they talk about how difficult anatomy is. Nationwide, I found anatomy has a pretty much a success rate of about 50%. And the thing I actually tell people who are trying to get into any sort of program is a lot of times people will tell you, oh, you need nursing or any health is that we're looking for people. And it is, but getting into the programs is a lot harder than most people realize. The reason is because while some programs will tell you that you need a minimum of a 2.5 GPA to apply, when you look at who's actually getting into these programs, you'll notice that some of them have a GPA of 3.9 for average for their incoming class. That means that for you to be competitive for programs, you need to get pretty much a 4.0 to be competitive. So I would actually tell people when you're taking this class, make sure you're taking it serious. There are 3 classes that are known to be extremely hard: anatomy, physio, and micro. So when you are studying, make sure that you have time to do those classes. And so with that, I will begin. And there are some questions I want you to begin to think about as I talk. One of them is, what is a sagittal suture? What a coronal suture would be, and what a transverse colon would be. And these will make sense when we get deeper into this. Now, when you're dealing with anatomy, there are basic terms that you see over and over again, and this language doesn't change. Now, I did not put in alphabetical order because I want you to be able to see certain things. For instance, the language, when you hear bi, it always will mean two. Lateral will mean to the sides. Sim means same, metry means measurement. You also do have some stuff like dorsal, ventral, medial, and uh, also thoracic and abdominal, which we'll talk about later. Now, if you look at this body here, we actually talk about things being the image here. We'll talk about how the human body plan has bilateral symmetry. So it has bilateral means it has 2 sides. So if you were to take a line and put it down the middle here, you actually divide it into even sides. So that's why we call it bilateral symmetry. We actually can also talk about things being either dorsal, which is on top, or ventral, which is on the bottom. Now this changes a little bit because of how we pretty much develop. So you'll see certain things like dorsal and ventral change a little. Median or medial will mean middle. Lateral means to the side, and that it'll make sense why I'm actually using these words first. Now over here you can see a line cutting this down the middle, which when a plane cuts a body directly in 2 sides, we call it mid-sagittal. Mid means middle, sagittal has to do with this, which is Sagittarius, and you see a bow and arrow. So sagittal is a plane that you would want an arrow to travel. Mid-sagittal is down the middle. Parasagittal would be next to the mid-sagittal. And anything that is closer to that midline would be referred to as medial. Anything farther from it would be lateral. Now, we actually would see the body in anatomical position. So lateral would be where the thumb is compared to the pinky. Medial would be the pinky compared to the thumb. Or in the chest, you can actually see the shoulder to the trachea. And we'll talk more about that later. So we also have a couple other vocabulary words, which I think is important for you to know. One of them is trans, which means across. So if you've heard of something being transatlantic, it means it's going across the Atlantic. A transverse cut is a cut in the body which is pretty much going across the body in this plane. It's going to separate things into different areas. For instance, if it's below, it's going to be superior. If it's above, it's going to be superior. If it's below, it's going to be inferior. And so we can actually look at superior and inferior. Usually this is mainly for the chest and head. When you get to the arms and legs, there are certain things that are a little different. And so when you hear a transverse cut, it goes across the body. So we've actually had a couple words that have come across: the sagittal, the medial, and the transverse, two different planes, the mid-sagittal plane and the transverse plane. Now, another cut we have is also can be called frontal or coronal. I like to use corona, and the reason I actually use corona BIRAD here is because corona means crown, which you can see here. And so if we cut something so it follows the crown of the head, a plane coming down here, it would be a coronal plane. Anything in front of the plane would be anterior. Ante means before. If you speak Spanish, antes. Behind is posterior. So you can see some things are being posterior and anterior. You can also see superior and inferior as well in this plane. Now, I asked a couple questions and I'm going to show you why this is important. If you understand what sagittal means, and you see a skull here, this is the anterior portion, this is posterior, you can see a line that goes down the middle, straight down from in a plane here. Because that plane would be a sagittal plane, mid-sagittal in particular. In a skull, when you have a suture, two joining that you can actually see here, that would be the sagittal suture. This one here goes with the coronal plane, and so that's the crown, the coronal suture. This one's a different one called the lambdoid, which looks like an L kind of, and we'll talk about that when we get into bones. But right away you can see why sagittal and coronal are such important directions. Now, I also mentioned about the transverse cut that goes across. Colon is the large intestine here. So if you notice, you have a part of the large intestine that moves across the body here. So that would be your transverse colon. It follows a transverse plane. Now, for the arms and legs, we actually do have something else. And the words I want you to use are proximal, distal, and we are using medial and lateral. So the thumb would be lateral, the little pinky would be medial. Same here. Now, if you look at proximal, if it's closer to the torso, that would be proximal. If it's farther from the torso, it'll be distal. So the shoulder would be proximal if you're looking at the elbow. The elbow would be distal compared to the shoulder. And I'll show you why that's important here. If you look at your fingers, which are called phalanges, you will see that there are two joints in the fingers. And if you pay attention to your finger, the two joints, anything, don't use your thumb, use any other finger, you'll have one that's closer to the wrist, one that's farther to the wrist. So one that's closer to the wrist would be actually more proximal to the rest of the body. So it would be the proximal interphalangeal joint. And the opposite would be the distal interphalangeal joint. Now, there are other words that are important. For instance, we actually talk about things being deep. Pretty much deep means it's deeper than anything else. Superficial would be on the top. So deep is actually is determined by looking at where the skin is going either in towards the middle of the body or away from the body. We also do have superficial, which is, like it sounds, superficial. Cranial would be towards the head. Vertebral would be in the vertebrae area. Abdominal would be in the abdomen, and pelvic would be pelvic area. This will actually make more sense as we talk more about it. Now, when we go into embryology, we'll talk more about this, but these two cavities do work with the dorsal and ventral. Dorsal, again, means the top, and you can see the area in here, and here, this whole area here would be the dorsal cavity, and in front of it, which would be a little lower, would be the ventral body cavity. The dorsal body cavity has a cranial meaning head, and the vertebral. body cavity. The ventral here has a couple different ones. You can actually divide it into the thoracic, the abdominal pelvic, which is then also divided into the abdominal and the pelvic. This gray line here represents a muscle called the diaphragm, which separates the abdominal pelvic from the thoracic. Over here, we can actually see the thoracic body cavity over up here. You can see the abdominal pelvic. You can't see the cranial up here as well. But what I wanted to show you is the thoracic cavity is divided into three parts. Two of them are the pleura, this one here and here. And the one in the middle is called the mediastinum. Again, if you remember, media means middle. So in the mediastinum, you would have the heart, the trachea, the esophagus. In the pleura, you'd have the lungs. In the abdominal, again, you have the abdominal and the pelvic region here. Now, I asked this question earlier, which is proximal interphalangeal joint, which is distal? You see the wrist here, and you can see, we're not going to use the thumb, we're going to use a second. You can see that in the fingers, which would be from here to here, you have one joint here, which is farther away, so this would be the distal. And this here would be the proximal interphalangeal joint. This joint here, these here, are actually known as the metacarpophalangeal joints, which we'll discuss more in bones. But you can see that you have distal ends and proximal ends of bones as well. Now, going to cells, there's a lot of things that I want you to know about cells. And one of the first things I want you to pay attention to is some of these words that I want you to actually come across. Some of these you should have come across in general bio. So for instance, the questions I want you to think about is, what would an endoplasmic reticulum be? What happens if DNA is mutated? What happens if a DNA mutation is in an area that is used temporarily when making a protein, but not part of the protein? Think about that. What would glycolysis be? And what is aerobic respiration? I will answer all these questions as we go along, but I want you to think of these. Some of these you have heard before in general bio. So again, there's always vocabulary that you want to know. And I actually have a cell here, and we're looking at a human cell which is a eukaryotic cell. And there are words that we need to understand so we can actually understand how things work. One of the first ones is hydro. Hydro is going to be water. So if you have a fire hydrant, hydro is water, so a fire hydrant is something that actually gives you water, and then there's a fire. Lipo. Many of you have heard of lipo, like liposuction. Lipo or lipo means lipid or fat. And so hydro means water, lipo means fat. And we have two words to remember. Phobic, which means afraid of. So if someone's arachnophobic, they're afraid of spiders. If someone is hydrophobic, they're afraid of water. Lipophobic, it's afraid of fats. And if you're afraid of something, you want to try to get away from it. Philic, phil, like Philadelphia or philosophy, means love. And so if something is hydrophilic, it means it loves water, which means it will dissolve in water. Hydrophobic won't. Lipophilic will dissolve in fat. And one thing to keep in mind is that cells exist in a mostly aqueous solution. Most of the things that they're in is going to be water. And so let's start talking about the basic eukaryotic human cell. There's a lot of things I'm going to just mention real quick, and I want you to think of what things are. So we have something called a cell membrane. The cell membrane is composed of phospholipids, phospho for phosphorus, technically in this case it's phosphates, and lipids which are fat. The phosphates are water soluble, lipids obviously are lipid soluble, and so you have a water soluble and a fat soluble end. They're hydrophobic on one side, hydrophilic on the other, and so they're going to react a certain way. In the cell membrane, you also have transmembrane proteins. Trans, again, means across, membrane, so you have proteins that go across a membrane, which are called transmembrane proteins. Peripheral proteins, just like it says, they're going to be on the periphery. You also will have nucleus, cytoplasm, endoplasmic reticulum. Now, if you look at this, endo means inside, reticulum is a net-like structure. So endoplasmic reticulum is an inside net net-like structure, which you're going to see in the cell in a little bit. We do have Golgi, vacuoles, cyto, which usually means cell, and skeleton, what gives you structure. So a cytoskeleton is proteins that give cells a structure, and we do have mitochondria. So here I actually decided to blow up the phospholipid bilayer, and you can see that you have these green areas representing phosphates, this green area representing phosphate here, and the lipid tails here. And as you see two levels, it's called a phospholipid bilayer. Over here, I decided to draw in a transmembrane protein and a peripheral protein. So you can see one of them goes all the way through, one of them is just sitting there. And these can act as enzymes and different things for the cell, channels as well. Over here we have a nucleus, and I made it yellow because it is composed of phospholipids. It does have transmembrane proteins across it, which are called nuclear pores. Over here I drew our mitochondria, which has a phospholipid bilayer outside and an inner membrane as well. This is actually going to allow you to take oxygen and the byproduct of breaking down sugar and other substances as well, and break it down with oxygen, called aerobic respiration. Now I also put the transmembrane protein, the cytoskeleton proteins. You can see the Golgi, vacuoles here, ribosomes here. Now, cells need enzymes to work. What an enzyme is it's a protein catalyst. And a catalyst is just something that speeds up the reaction. Usually, enzymes will have a ending which is ASE. So anytime you see ACE at the end of something, it's going to usually be an enzyme. For instance, over here, I actually wrote a sodium, Na for sodium, K for potassium, ATPase. It's a protein that takes ATP, which is adenosine triphosphate, and breaks it. And anytime you break something, you end up getting energy. And that energy is used to move 3 sodium out and 2 potassiums in. And so you can use these proteins as catalysts using enzymes, breaking it, and using that energy to move things against a concentration gradient. Now, there are two things to know. When you're actually breaking stuff for energy, it falls under catabolism. When you're trying to build stuff, it's anabolism. Anabolic steroids, for instance, are steroids that help build you up. Catabolic steroids will actually help break down the body. And the way this works in a basic level is you have DNA, which is going to be found in your nucleus. The DNA has the code for every protein you're going to need. And it's going to be copied into RNA. RNA is ribose nucleic acid. And that RNA will leave the nucleus through pores. And it will bind to a ribosome. Ribosomes are enzymes in many ways. Organelles, sometimes people will say it. And one of the things you'll notice with ribosomes is it has RNA is ribose nucleic acid. Ribosome has the same ribose. Ribose is a sugar. So ribosome works on ribose RNAs. And what happens is it reads 3 RNA bases, and that encodes for 1 amino acid. And so you make these proteins 1 amino acid at a time reading the RNA. Now, a lot of times you're not just making one protein. You can have multiple ribosomes bound to it, so you can end up getting that. Now, if this protein is just going to be used inside the cell, and it's not going to break anything inside the cell that you don't need broken, then you can actually make it in these ribosomes. But if this is a protein that's going to be excreted or is going to be put in a phospholipid bilayer, the ribosomes will bind to the endoplasmic reticulum, forming a rough endoplasmic reticulum. It's rough because it has ribosomes. It's going to make the proteins in that, put in little vacuoles, send it to the Golgi, and the Golgi then modifies it as well and puts it in vacuoles to decide whether it's used for the cell or is going to be excreted. So proteins can be released. Proteins can stay here and be used for different things. And so there's a lot of different things that can happen. Now, I talked about catabolism, and that's how the cells get energy. And I want you to pay attention to certain words. For instance, sugar The sugar that we tend to use is a 6-carbon sugar called glucose, and it's broken down for energy in the cytoplasm. Usually means bag, so cytoplasm is bag fluid. It's a fluid inside the cell. And when you take glucose and you break it into two, you make two pyruvates. Now, breaking sugar, one glucose into two pyruvates, is called glycolysis. Glyco means sugar. Anytime you see glyco, it means sugar. So if you hear something called a glycoprotein, it's a protein with sugar. Lysis means to break. So glycolysis means you break sugar. And you break sugar to get energy. Now that energy, you end up just getting 2 ATP from that, and the pyruvate can then end up being made into acetyl-CoA and shuttled into the mitochondria. And in the mitochondria, it can be broken down. Remember, it's 6 carbons each glucose. It's broken down on 3 carbon rings. And it'll be broken down completely in the mitochondria to carbon dioxide and water. And as you're using oxygen to break that down, it's called aerobic respiration. Glycolysis is anaerobic. An means without. Now, pyruvate can be converted to lactic acid if you're lacking oxygen, and that will temporarily give you a little bit more ATP, but it's going to have to be worked on later. Now, one of the neat things that happens is if you have too much acetyl-CoA, you will end up converting it into fatty acids. And so that's why when people's caloric intake is high, it doesn't matter if it's sugar, protein, as long as you're building up acetyl-CoA, the acetyl-CoA will end up building fat. So there are a couple basic cell questions I mentioned. And one is, what is the endoplasmic reticulum? So you can see endo means inside, plasmic, the cytoplasm. So inside the fluid, reticulum, it's that net structure. So it's a tubular net structure inside a cell, which has, in this case, composed of phospholipid bilayers. And it does a lot of different things, which we'll talk about more as we go into the different modifications of every cell. And what would, like I told you, it's like DNA is made one protein at a time, and a lot of times the DNA is used to actually make temporary structures that are cut away. Now, sometimes you can have a mutation in the temporary structure of the DNA, and it will not affect the final product, but it could change the way it folds sometimes. So if the DNA mutation is in an area used temporarily when making a protein, but is not part of the protein, sometimes it's what's called a silent mutation. It's not seen. It's when it's modified. Glycolysis. Glyco means sugar. Lysis means break. So glycolysis is a breaking of sugar. And when you break sugar, you don't use oxygen. So it's anaerobic respiration. And what is aerobic respiration? It means that you're breaking something, usually pyruvate, using oxygen. If it's not using oxygen, it's anaerobic. Now, I am going to mention about how the body is built. And this is actually something we can go relatively quick in. The basic unit of life is the cells. And cells are composed of organelles, and organelles are composed of proteins, lipids, and carbohydrates. These are actually made of different, pretty much different atomic structures, which you learned in general bio. Now, cells come together and they modify based on how these things are going to be used. And so what you end up having is tissues. Now, there's 4 basic tissue types which have a bunch of other subdivisions. When you are asked what type of tissue it is, usually we're not talking about is it epithelial, connective, contractile, or nervous. We're actually wondering if you can tell us what the tissue is, not just in the 4 tissue types, but in the more specialized one. Now, usually epithelial, endothelium, and mesothelium will line the body and usually has a bit more cells than matrix. It usually has cells in a certain area, and the matrix really is a thin layer of proteins that hold it in place, called the basement membrane a lot of times. Connective tissue, as the name suggests, connects the body, and this usually has more cells than matrix. The difference is adipose, because adipose tissue, fat, will have a lot of its matrix inside the cell. We also do have something called contractile tissue, and this is the type of tissue that causes movement. Now, there are sometimes people talk about when a wound gets cut, when skin gets cut, it may contract. We're not talking about that type of contraction. We're talking about active contraction. The other one is just tissues pulling away from an area. And nervous tissue sends electrical messages. Now, tissues, like I said, you have multiple different types. They come together to form organs. So you don't have heart tissue. In heart tissue, you will have a endothelium tissue, which lines the inside, mesothelium, which lines the organ itself. You can end up having connective tissue, which gives the heart structure in a certain way. You can have contractile, such as myocardial tissue, striated cardiac muscle tissue. And you can have nervous tissue, which sends messages to either slow or speed up the heart. And so organs have various tissues. Now, just like the heart, it doesn't work alone. You have a cardiovascular system, you have organ systems, and all the organ systems come together to form organisms. Now, how does everything begin? Well, for us to look at it, we actually want to look at mitosis. Mitosis is a normal cell division for normal cell growth and to make or repair tissues. But usually these cells are not constantly dividing. They're usually in a stage called a G0 phase, which is the cells are just alive, they're doing their normal things, they're not dividing. When they want to divide, the cells have to have their DNA copied, and they're held together in chromosomes. And these chromosomes have to be copied before the cells can divide. Now, human cells have 23 pairs of chromosomes, so I usually tell people don't necessarily try to draw every chromosome because it'll drive you insane. Instead, understand that there's 23 pairs of chromosomes, and of those 23 pairs of chromosomes, 22 are autosomal, and the last two are sex. The truth is, sex chromosomes, only females have two X chromosomes. Males actually don't have two paired chromosomes. You have an X and a Y, and there's a little difference. Now, there is a bit of a difference when you're actually looking at this when you see a little n and a big N. Usually it has to do with what we're calling a chromosome at that moment. And when we're dealing with chromosomes, we have a lot of different things we want to talk about. Ploid is a genetic makeup. So there are two types of cells. We have diploid cells and haploid cells. Di means two. So diploid is cell that has two copies of its genetic makeup. Haploid means half. So if you only have half your genetic makeup, saying you only have 23 pairs of 23 chromosomes not paired, you'd have a haploid cells. Now, chromosomes are how DNA is held together in the nucleus. And you do have chromosomes that can have two copies of a chromosome, which are called chromatids. And so this is how it works. Most cells will be in something called interphase. They're going to be diploid cells, and when something triggers to divide, they actually have to make the cell a 2 big N. If it has to divide, it then goes through mitosis, which is divided into a bunch of other groups. Now, these are not one and then the next one. These are phases, so you can't have early prophase, mid prophase, late prophase, early metaphase, and that. But here I'm actually going to go simple. So in prophase, pros means before phase, the first phase, you're trying to get the cell ready. And so what ends up happening is, in interphase, before the cell divides, the chromosomes are not really easy to see. You'll probably see a blue nucleus based on the staining, but you're not gonna be able to make out the chromosomes. In prophase, the chromosomes condense so you can see individual chromosomes. The nucleus is broken down, and you start making a special protein called mitotic spindles, and these are going to attach to the chromosomes. Now, once they're attached to the chromosomes, the mitotic spindles, notice the name mitotic, it's telling you the spindles you need for cell divisions. they're going to take the chromosomes and try to line them up so the homologous chromosomes are next to each other. And when the mitotic spindles line up the chromosomes in the middle of the cell, we call it a metaphasic plate. So when you have all the chromosomes lined up in one area, we call it the metaphasic plate. Then the chromosomes are pulled to opposite ends by the mitotic spindles. The spindles actually shrink, and so they pull the chromosomes. And when you start pulling the chromosomes away, we call that anaphase. In anaphase, homologous pairs of chromosomes are pulled to opposite sides of the cell. And this ends when you start forming a nucleus, when you form a nucleus. Homo means same, so homologous means they're relatively similar. And then you end up having telophase. Telophase is the cells moving away from each other. So you would have two nuclei and the cell would start reforming and moving away from itself. It's called cytokinesis. Cyto means cell, kinesis means moving. So the two daughter cells move and split, and now you end up having two cells. This is important for embryology because what we start with is we start with a giant diploid cell which divides rapidly in something called cleavage. It's going to end up forming a collection of cells called a morula. So here, I can't remember where I found this picture, but it's very useful. And so this cell divides and actually ends up forming this big ball cell called a morula. This is another fair use picture. Actually, I can't remember where or how I found it, but the morula ends up forming something called a blastocyst. Cyst, in this case, doesn't mean cell. It goes back to meaning bag. And a blastocyst has two distinct regions. You have the trophoblasts and the inner cell mass. The area that we're most interested in is the inner cell mass because it's going to form what's going to end up being a human at the end. Now what ends up happening is in week 2, you'll notice you have two cell layers, one here and here. And these cells form these spheres. It's actually in a sphere. And so this line here is a disc. As there's two cells, we call it a bilaminar disc. Bi again means two, laminar, laminate, layer. So it's a two-layer disc. Now on the top area here, you have what's called the epiblasts. Epi means outside. On the bottom area, you have hypo, which means, epi means top layer. Hypo means lower layer. And so what you end up having is you end up having a fluid-filled cavity here called the amniotic cavity, which is filled with amniotic fluid. And where the hypoblasts are, you have a fluid-filled cavity called the yolk sac. So remember, epi means top, hypo means bottom. And then we end up having week 3, the trilaminar disc. And what ends up happening is the cells from the epiblast come into an area between the hypoblasts, and so you end up having 3 cell layers. At that point, you have an ectoderm. Ecto means outside, derm means skin. The mesoderm, meso means middle, derm means skin, and endoderm, which means inner skin. The reason it's called that is you're going to end up forming a notochord in the mesoderm, which is going to cause a pull on the ectoderm, forming a neural tube. That is going to be your nerve, spinal cord and brain. So when the body develops, what you end up having is you end up having the outer layer come in here, forming a tube. And this tube would be your neural tube. Now we have a dorsal and a ventral. This tube here would be the endoderm, which would be your gut tube. Now I put this same picture that we have before, and I colored the cranial cavity and the vertebral green, just like in here. And I put the ventral cavity blue, just like here. You also will see that the cavity is here and here, and it is going to be covered with a mesenchymal membrane on here, which is going to give you other aspects. And you're going to have mesoderm around this area forming internal organs. And so with this, we can actually start talking about the different tissue types. We have epithelium, like I said, it's usually more cells than matrix. It's found lining in body cavity. If you think of skin, for instance, the outer skin has three layers. The outer layer is made of a epidermis, which is epithelial cells. The layer underneath, the dermis, is a connective tissue. You do have some contractile tissue within skin, which helps it contract, and you have a couple other connective tissues as well, and you have nervous tissue in there. So the tissue types you have are these four, which we're going to get into a bit more in the next lecture. I hope you enjoyed this and have a nice day.